Hello and welcome to our webinar titled Breaking Ground in Direct Compression Tablets with STAB. So this is a brand new product from SPI and we're excited to bring this to you today, and thank you for joining. Today's presenters are myself, Wayne Camarco. I work in the technical development area at SPI Pharma, and also joining me today is my colleague Shivkumar Mahajan, and he's based in our Bangalore lab in India as a senior research scientist. Today we'll go over STAB as far as the introduction to the product, what it is, why we think it's needed, its physical properties and composition, flow, tableting, and some tableting studies on case studies, different APIs, and also a high-speed tableting run, and stability considerations, and then finally a Q&A. Before I go into some of the properties of STAB, I want to give a little bit of background about SPI. And on this slide you can see that we are actually part of Associated British Foods, ABF, and of that you might know companies like British Sugar, on the retail side Primark, and then part of that group we are in the ingredients area, which is ABFI, and you can see us down here as SPI Pharma, with some sister companies that you may know of as well. We're actually based in Delaware, USA, with plants in the US, but also R&D and plants in France and India, and a global distribution network. Within SPI Pharma, we do have a large range of products. We are actually a market leader and supplier of immediate release antacid actives. And we do specialize in patient-friendly type of dosage forms, patient-centric. On the excipient side, we are pretty well known for our products, orodispersible, chewable, ODTs. And as far as innovation goes, you know, as today, the new product we're coming out with today, we've got some more in the pipeline. And we can also offer customers prototype formulations on any of our platforms to help them get a little bit further ahead of their initial formulation. For today's webinar, we're discussing STAB as a platform to make a non-orodispersible tablets by direct compression. Of course, DC is generally preferred for many reasons, and on this slide we're showing some of the reasons. For example, shift from wet granulation to DC process, minimalizing the use of organic solvents, and of course reducing process steps and improving output, maybe freeing up some wet granulation equipment for other products which require it. Also nowadays things like spray drying of actives or engineered actives are more prevalent. These are often solid solutions or combinations of API or APIs with excipients, and those engineered products don't really do well mostly with wet granulation processes, solvents or water. So these are better suited for direct compression. So again, a reason to use that type of technology. And of course, focus on efficiency, and with the emergence of high-speed tableting, which is becoming more and more prevalent with companies using that equipment the best possible way, the best efficiency, maybe the highest speed that the press can offer, is obviously useful. And in continuous manufacture as well, direct compression plays its part. So what is STAB? It's a platform formulated to optimize tablet production as an all-in-one ingredient. So really you add API and lubricant and you can go ahead and make tablets. Well, we wanted to make the formulation simplified for formulators using such a product. It has excellent properties as far as tableting, and it'll be a quick way to get you to market. Now, we call it a pre-formulated product because it has everything ready to go. It is based on mannitol and silicified MCC, but is composed entirely of compendial excipients. And we've left out a few of the excipients that can have some issues with compatibility to give a little bit more flexibility with the API you might be using. And we're going to look at the robust tablets you can make. It flows very well and gives some good stability protection. And the table here gives some basic physical properties. So the D50 is about 133 microns. But I think what we want to look at more is flow. So, of course, flow is critical for these types of materials. Something that's going to be dependent on the API you add. But, of course, you want that flow to be something as a really base property so that you can get good compression, good tableting. And here we're looking at some flowability measurements. On the left, we see the Flodex. Like this machine, it's a piece of apparatus which basically has a funnel. You put the material measurement into that funnel, and that flows into a container. And those, you have interchangeable metal discs. You can see them there, which go into the bottom of that container. And there's an aperture of varying millimeter sizes. And you generally start to see where you can get the flow through the smallest aperture, and that's in millimeters. So if we look on the right-hand side, we can see some of the products used to compare. And we like this equipment because it seems just like a tableting press would work as far as feeding material in. So you kind of got like the hopper, you're feeding into the hopper itself and actually into the force feeder, and then into the dies themselves, into the orifice of the dies to make the tablets. And of course, that's what you want to have: good flow, good content uniformity for the tablet weights. And flowability measured by this equipment is shown on the right. So S-Tab is really good. It's 4 millimeters, pretty much the smallest you can go on the aperture. Now, we're also looking at things like high flow lactose, simplified MCC, and the far right we've got MCC 102, some common excipients. And here we've also got Platform 1, Platform 2, and we'll be referring them throughout the presentation. These are two similar products on the market, which are all-in-one type of products. And so we wanted to compare also against those materials. To test the flowability a little bit more, perhaps under more challenging conditions, we added some fine ingredient as an API, like a model fine material. It's about 74 microns. It has a really bad flow. Car index is 39, which equates to very, very poor. So it's a good model to use for as far as putting APIs into the platform. What we've got here is a series of S-Tab versus Platform 1, and we're looking at 0, 5, and 20% addition of this fine material. And what we did is go ahead and see as far as the tableting and flowability, how that worked. And I think, obviously, with no material at all in there, the left two, there was fine, excellent flow. And but what we saw, once we put 5% of this material into the blends, the different blends, that there was problems with flowability with Platform 1 with this material, and some inconsistent blend entering the feed frame we observed. When we took that up to 20%, then it essentially was really not working at all for Platform 1, but S-Tab was continuing fine. And that's reflected in the bottom table, where we're looking at some of the standard deviations of tablet weights, which tend to increase with the lower flowability. So S-Tab here is going to can carry about 20% of this fine ingredient and retains good flow. As with many tablets, there's usually or can be a coating step. And so we wanted to look at our platform as far as making tablets and coating them to make sure that the coating process worked okay. And we're looking at placebo tablets with two different polymer-based film coatings at 3% weight gain. And we can see a little bit of increase in hardness and disintegration time, which is normal for film-coated tablets. But we see no defects, and the coating looks well on both polymer-based systems, no blemishes or surface imperfections. In the next few slides, we're going to look at a couple of case studies using drugs with different challenging properties. And we're using these as models which could model the type of properties found with other APIs. For the first API, it's furosemide. We chose this. It's a micronized API. You can see the type of structure, very small particle size. We're looking at tablets about 20 milligrams in a 250 milligram tablet, so about 8% containing in the formulation here. So we're using furosemide here. It has a low and poor flowability, so it's a good model for those types of APIs. And we can see here we're using about 8% by weight in the formulas there. And we're comparing it with a couple of the other platforms that we mentioned before, Platform 1 and Platform 2. This lubricant is added to the S-tab. We chose not to add lubricant into the product. We thought it would be more flexible for the formulators to add that in. Because if you already have it in there, sometimes that might, if you need more lube, you might want to use a different lube, so you're adding it anyway, or mixing lubes. So you could actually switch between such fumarate in our case, or mag stearate without a problem. So this is a use case example of the formulation type. And here we've got 3 different compression forces used for each of the platforms, and we're making tablets and observing some of the properties there. I think as far as the flowability, we see again the problem with Platform 1, the very poor flowability into making the tablets, and this actually caused the content uniformity as far as the weight. The content uniformity to be a lot higher. We think because of that poor flow. The hardness, the graph on the left below, the hardness, compactability of the S-tab is equivalent to Platform 1, but lower with Platform 2. And on the right-hand side, as far as disintegration, again, similar, slightly higher results with Platform 2. We also wanted to look at the drug release of the formulas. So here we're looking at USP dissolution. And we can see on the graph below the drug release profiles of the various platforms. For Platform 2 and S-tab, there's a good release, and maybe a little bit faster release for the S-tab and the earlier part of dissolution, probably explained by the faster disintegration seen in the previous slides. As far as the Platform 1, this does contain MCC, and there's a leveling off of dissolution, which can be explained by adsorption, and the reference to this type of effect is given on the right-hand side. So summarizing the use case with a poorly flowable API, we see really good flow, and we have some good flow versus one of the other platforms. Compressibility is good. Less API interaction we saw from the dissolution side of things, and then good disintegration. Also wanted to add on the bottom right of this slide that we do offer pharma solutions, I mentioned it a little bit earlier, where we'd like to offer to make prototypes of APIs that you might have using this product, or indeed any of our other platforms we use. We can work with you to make those for you for your early evaluations and get you a little bit faster to market. Looking at our second use case, we're looking at a drug which has some drug interactions with excipients, and this is diphenhydramine. And some of those stability issues Shivkumar is going to cover later on. But here, just giving some of the basic properties, particle size about 225, using a 25 milligram dosage tablet at a weight about 250 milligrams, so about 10% weight. As with the previous use case, we're looking at the tableting as far as the study and the types of properties we get. The shown here, we have S-tab and Platform 1. Platform 2, when we tried that, we couldn't actually use that in the end because we had some incompatibility issues. Again, Shivkumar is going to go into that a little bit later. But looking at these two materials, we see good tableting properties. But we do see a problem with flow, again with Platform 1 using this material, 25 milligrams. And we saw that as flow going into the hopper. We did see some content uniformity higher on the higher side. But interestingly here, which really stuck out is the disintegration time. Really goes very high with Platform 1 and up to plus 1,000 seconds. And summarizing, we were able to make good tablets here. We did have issues with platform 2 with some of the stability indicators. But STAB had a good flowable blend, and we worked very well as far as the general properties. And we had good accelerated stability also with this product. Again, I'll go into that a little bit later. So just to say again, and to emphasize, we can help you with early formulation if you would like to look at one of our APIs, or us to take that and to make some prototypes for you. Here we're looking at another use case we added, looking at nutraceutical model, and actually it's a pomegranate extract. It's spray dried, and you can see the tablets made from it on the right-hand side with the coloration. I think it's nice to look at this. It's a spray dried material, as we say, a nutraceutical, and we made good tablets with STAB here. We did compare it with the platform 1, and what we did see is very high disintegration times. In fact, 2.5 kilonewtons. It was 17 minutes, and we didn't measure after that because it got longer and longer. So it's acceptable tablets made with STAB. And I'm going to now hand over to my colleague Shiv Kumar now, and I'll finish off at the end of the presentation.
Thank you very much, Wayne. Hello everyone, and welcome to today's webinar on STAB. I am Shiv Kumar Mahajan, working as Senior Scientist at India Development Center of SPI Pharma. I have more than 12 years of experience in formulation development. I will take you through high-speed tableting, then we'll see stability considerations of STAB, then we'll move to last part, that is conclusion, and in the last we have a separate Q&A session for the audience. Here you are able to see the high-speed compression details, where we have used HATA CVX 45 station for compression of the STAB, along with 1% lubriform. The punches we have used is 6.5 mm round shape punches. Target weight we have set in some trials is around 85 mg, and some trials we have targeted around 90 mg. Those data we are able to see in a couple of slides. If we talk about the production rate at 60 rpm, from one side we are able to get 1,62,000 tablets per hour, and from both sides it will be around 3,24,000 per hour. So we have used around 30 kg of material, but our aim was to run the machine as longer as possible. So that's why we have run this compression machine for only one side. We have done also the sampling for 15 minutes, every 15 minutes till the compression run of 2 hours, so as to check at every 15 minutes how is the weight variation. So we are able to see the weight variation of tablets, individual tablets, and even the group weight of the tablets. You are going to see the high-speed compression trial video after 3-4 slides. But before that, let me take you through the PLC of the software used by HATA. What the PLC says, what are the components of the PLC. After 3-4 slides, then we will be able to see the video of high-speed compression of about its PLC, what is the software is telling, and what are different components at PLC you are able to see. So this is the first screenshot of the PLC, where you are able to see on left hand side the turret RPM is there, feeder RPM. On right hand side you are able to see how many number of tablets of compressed at that particular time point. Then the number of rejected tablets. On the bottom you are able to see the main compression, where there are 45. This is a 45 station compression machine. So the standard deviation within 45 punches is given here as a standard deviation, which is 0.22%. And here on the lower side there is bars in different colors, green and yellow, which talks about whether they are within the set limit or within the adjusted limit. In this screenshot, not much, only how is the feeder setup, what are the parameters of the feeder setup that is being shown here. Here all the compression forces details are given, pre-compression, main compression, ejection force details for all 45 punches of B side. This black line is force trend data, and we are able to see it is very much stable throughout the 2 hours of compression. This tab on PLC is for auto weight control parameters. So during video you are able to see this white cursor is moving near to the target throughout the 2 hours of compression. We have captured this high-speed compression video and divided into 2 parts. In first part, you will be seeing the PLC components, and in second part, the actual machine, how it is running and how the tablets are coming. So let's see the first part of the video. Let us see now the remaining part of video where S-tabs compression at 60 RPM is going on. You are able to see how the movement of punches is going on and how the tablets are coming out from the machine. As we have seen the video of high-speed compression trial, where we have seen the consistency of data throughout the 2 hours of compression with respect to main compaction force, ejection force, the trend line of it. There are very low number of rejects observed even after 2 hours of compression at 60 RPM. So let's see now more detailed data of high-speed compression tablets. So here we have done multiple trials with S-tab at high-speed compression so as to check the consistent performance of it at high-speed compression. So here one of the best data we have kept where the target weight was set around 85 mg, and in all 3 graphs you will be able to see the average mean weight is exactly at the target, and the individual target tablet weight variation also it is within plus minus 2 to 3 mg. Even we are running at very high speed and the production rate is also high. So here also we have seen the same main compaction forces around 0.2%, and at all the time points what we have done the sampling at every 15 minutes throughout these 2 hours of compression, the very minimal weight variation was observed. So based on this data, we can conclude that S-tab is best suitable material for high-speed compression purpose. We also check performance of S-tab against highly flowable lactose on same compression machine HATA at same speed 60 RPM. This time we set target weight around 90 mg, and we have used scale-up batch of STAB for this compression. So on left hand side you will see weight variation for highly flowable lactose is high at every time point from 15 minutes to till hours. It's very high compared to STAB. So in STAB at each and every time point you will see the weight variation is within very narrow range throughout the 2 hours of sampling. On bottom, when we see individual tablet total weight variation against time, here also highly flowable lactose is on higher side, whereas the STAB is on lower side. So when we compare number of rejects with STAB, we found 0.14% number of rejects, whereas with lactose we found around 1.1% of reject. Again, when we compare all the points, minimal weight variation and number of rejects even very low, STAB's performance again here better than the highly flowable lactose. Based on the data what we have seen in previous slides, here is the conclusion of high-speed compression trials. Individual weight variation found to be extremely tight compared to DC lactose. Number of rejects found to be 6 times lower than DC lactose. And by using STAB, significant amount of cost saving can be done while a formulation is having high cost actives. And even while we are running at very high speed, productivity is high and the rejects are low, so the yield will be high. So that's how we can sell more number of tablets to the market. Let me take you the observation from machine supplier when we are performing these high-speed compression trials at their site. The observations are like when they are performing many trials of products produced through weight granulation process, they have never seen such a low standard deviation of within punches, which is less than 0.5%. Mostly they have observed around 1.5%, but with this is the only material they have seen the weight variation within punches around 0.2%. And this is very surprising from them. And they also told the performance of STAB even at high speed, this much high speed at 60 RPM. And when we are setting the very low tablet weight, that is less than 100 mg, 95 or 90, the weight variation is also very tight. So again, that was surprising for them. Moving on to the next part of webinar, stability considerations. In this, we are going to see stability data of STAB as placebo, STAB with model drug diphenhydramine, and risk assessment of STAB against platform 1 and 2. Stability study of STAB platform done in 2 forms. One is in tablet form and second one is as such blend. So packaging details given on the left hand side. First table you are able to see accelerated condition 6-month data of tablets in which hardness, DT and friability is almost similar to its initial results. So no significant change observed in those results. At the bottom, when you see the results of blend, in this also up to 6 months, there is no change in all the test parameters, whether it is BD, TD, or even LOD. At initial it is 2.7%, whereas after 6 months it is 3.4%. No significant change observed. So overall, we can say based on all this data, STAB is stable at the end of 6 months of accelerated condition for both the forms, whether it is tablet and even for blend form. Test stability data by using model drug diphenhydramine. As my colleague Wayne has already taken you through the case study of diphenhydramine. The same tablets were subjected for stability study at accelerated as well as long-term stability study. So here we can see the hardness is slightly increasing, but it is not having any impact on DT. It's less than 1 minute. Similarly, friability is also not increasing, so that there is no softening of tablets. With respect to assay, water content, and dissolution, these are all meeting its acceptance criteria. There is no significant change with respect to initial. And with respect to related substances, none of the impurities out of limit, or even it is not increased from its initial, even after 6 months of accelerated condition. So based on all data, we can say STAP is pretty stable with any kind of actives, or even with can be used with wide variety of actives which are very susceptible for oxidation or any kind of degradation. With most of the pharmaceutical actives, oxidation is the second most common degradation pathway in it after hydrolysis. Oxidation is more complex and hard to control for formula esters. As we know, oxidation will happen with 2 to 3 different mechanisms, whether it is auto-oxidation, peroxide mediated oxidation. Here I have given some of the examples of the molecules which undergo oxidation or which are prone for oxidation reactions. In the bottom, we are seeing 2 tables. Those are taken from one of this article. In that, we are able to see there is generation of N-oxide impurity in presence of peroxide with raloxifene. And the right hand side, with different excipients, the amount of N-oxide impurity generated is different. It's showing more with povidone and cross-povidone. So some of the excipients which are having a high number of peroxide with them, so those are causing more N-oxide impurity. And the second part is high potential for N-nitrosome impurity. So some of the excipients again are having high nitrite contents with them. So those are also a risk for N-nitrosome impurity during development of a formulation. To show relative tendency of STAP for oxidation, we completed a comparative open exposure study by using diphenhydramine as a model drug at accelerated condition for 61 days. Here are the results of the after 61 days. So you can see with STAP, N-oxide impurity is around 0.01%, whereas with platform 2, it is 0.19%. So significant amount of difference we have seen with platform 2. So based on these results, we can conclude that STAP can be used with wide variety of actives which are prone for oxidation degradation pathway. When we do the stability risk assessment of all 3 platforms, the STAP, platform 1 and platform 2, the stability risk associated with platform 2 will be on higher side because of its main components like highest concentration of cross-povidone, which leads to high concentration of peroxide content in it. Even that's why it is more sensitive to the oxidation reaction, and same what we have seen in previous open exposure study of diphenhydramine. Again, when we compare all the excipients and their nitrite concentration, cross-povidone contains highest concentration of nitrites with them. So the risk associated with N-nitrosamine impurity again on higher side for platform 2, and for STAP and platform 1 it will be on lower side. So overall, when we consider and main components and when we do the risk assessment for all them, platform 2 has high risk, whereas STAB and platform 1 are lower risk. With respect to platform 1, even though it is stability-wise on low risk, but when we have seen the previous section with vein, the poor flow of platform, that's why it's not even coming from hopper to the feed frame of compression machine. So overall, with respect to stability, flow, STAB is well-balanced platform for formulators.
Thank you, Shivkumar. We'll address some questions next, but in conclusion for this presentation, we've talked about some advantages such as superior flow and stability, so allowing use across a wide range of APIs, and more operational flexibility. Also, as an optimized pre-formulated product, we can deliver faster development times and faster production rates. For example, we've seen with high-speed compression and also reduced rejects. And if you can keep those numbers down, especially importantly for expensive APIs and not throwing those away, that's a great advantage. And finally, as mentioned, we can help you with any prototype developments. Now to the Q&A session. Got a few questions here. Shivkumar and I will answer. First question is, can you comment more on the various approaches to measure flow? Yes, actually, good question. Commented a little bit earlier in the presentation on this, but of course, as you know, there's many ways to characterize flow, angular repose, something like density-based, such as Carr's index. However, we think the Flodex has the advantage can potentially mimic the operation of the feeding of a blend from a feeder into a die. And we actually found good correlation with the Flodex results in our tableting runs. And that was both at the lab scale and at high-speed tableting.
We got one more question. What are the benefits of STAB or available market products? So STAB is a well-balanced platform which gives best flow, good compactability, DT with low risk of stability. Against platform 1, where we have seen, which is a MCC-based platform, it is having very poor flow, even though it's having high compactability, but it's failing at some of the examples like diaphenhydramine and nutraceutical actives. It's failing at high main compression force and DT. So higher compactability again not beneficial at every point. When we compare STAB against platform 2, it is having good flow but inferior compactability and high risks associated with stability. And for DC compression, most important criteria is flow of the material. So we have demonstrated it at high speed compression against the best flowing lactose in market. So these are the benefits of STAB over the available market products.
Okay, I can read one out as well. What is the max drug loading you can use? We did have some information on some of the charts there. Generally about 10% by weight is what we were looking at. But the 25% we did use for the Nutra example shown, and in fact we did actually use 25%, a much higher amount in the diphenhydramine. It's not shown here, but it worked okay as well at that higher amount. And of course any usage levels are quite difficult to predict. It's going to depend on your API properties. Maybe if it's very, very low compressibility, that'll affect it. Or perhaps a sticky or tacky API where you might need to use a little bit less material.
We got one more question. I just read it out first. Can you give us some examples of specific actives you think would be benefit from being formulated with STAB with its rationale? So I would suggest some high costly actives like entecavir, brexpiprazole, eloxaban, vardenafil. So by using STAB, as number of pages will be less, so cost saving can be done with these highly costly actives. And some of the actives which are having RS sensitivity, those can be developed with STAB. For example, tenofovir, methotrexate. So those APIs can be used for formulation development with STAB. Thank you.
Okay, well, this wraps up today's webinar. There were some additional questions submitted. We didn't get time to answer all of them, but we'll get back to those who submitted. If you would like, of course, any samples of STAB, please, we would love to send you some, so let us know. And I think in conclusion, thank you very much for attending today. We look forward to having you at our future webinars. And from Shiv Kumar and myself, wish you the best for the rest of the day. Thank you.